So, we were with that extradiol aromatic one. So, if we have a extradiol aromatic ring, how we can clip that? So, these are the cleavage dioxygenases. So, we consider them that as extradiol aromatic ring cleaving agents. So, they are the very good reagents in that way, and the ligand environment we are talking about is 2 His 1 carb type and is basically a facial one. So, we can consider it as a facial triad. That means you have 3 donor atoms and there are a large number of such enzymes available, and at least 20 families have been identified so far. Of enzymes and simply we have seen that if we have that iron and immediately we can have the octahedral site, 2 of them will be occupied by nitrogen and 1 by oxygen and the facial orientation, and that we get after if you have the site, the catalytic site, and then the catechol is binding there. So, you have the catechol there, so it will bind these 2 sites. So, 5 positions are occupied now, and the same simple strategy that we will take or we will take the help of dioxygen molecule, and that dioxygen molecule will now go and bind to that particular available site, that means the 6th coordination site. So, straightway we can write all these positions. So, you have the nitrogen, nitrogen, oxygen, then O, this is also O, and interesting thing happens here onwards, that if your center is getting reduced, that means your bound ligand should be oxidized. So, if we have these 2 positions like this, so we have a oxidized catechol site, and if that oxidized form is there, we know that there are 2 other forms when we go from catechol redox process. The first one is the semiquinone and the second one is the quinone form. So, single electron oxidized form is coming into the picture, and you have the binding of this dioxygen. So, dioxygen is simply bound over here, and this is also reduced. That means, this dioxygen is going to the corresponding superoxide species. So, the change in the corresponding status for this dioxygen and the change in the ligand environment basically giving something where the reaction is mediated basically through this iron site, and that can be very easily identified if we just simply use some 18 oxygen. That means the labeled oxygens are used sometimes. So, 18O labeled one is used. That means we want to have a track for the dioxygen one where it is going. So, initially it is bound to these two positions, then the binding thing is changing towards the semiquinone form. What is there? So, this is one and this is the original oxygen. That means this position is getting activated, which is the radical center which we generate from here. So, this position is activated and this dioxygen molecule will come and attack this particular position. So, once it is like this, so this dioxygen still bound to your iron site, and this one will also have a typical coordination from this oxygen as well as this oxygen. So, we all the time we are living with something where we are just talking about some facial positions. So, all the three positions, that means N, N, O are still occupied by the protein chain, and we are doing something on the other three positions. And these oxygen, so these two oxygens are the new dioxygens which can also be labeled one. And this particular arrangement is not so a stable arrangement in a particular organic molecule when it is attached to this particular benzene ring. So, it has to be stabilized. So, the second sphere of interaction is from some imidazole nitrogen which is available from the protein chain. So, this particular arrangement, because you have a charge already on O2 and that charge is getting stabilized through the hydrogen bonding interactions with the imidazole hydrogen. Then in the next step you have the ring opening reaction, and that ring opening will give you a 7-membered ring with this oxygen inside. So, this was still bound to your iron. This oxygen is there, and this O, if you consider these two, will be now your OH bond. So, now you will have one OH group attached to this iron. So, you are leaving behind the other three positions. Then at this last step you have the ring opening now. and these are your labeled oxygens. Here the labeled one is coming, that means we are able to insert both the oxygen atoms which were available there, and still the open form of the catechol, still it is bound to your iron site. And lastly, at the final step, our product is released from iron coordination, and thus we get the extradiol cleaved product, which is CO2H So, the interesting part of the entire reaction sequence is that the particular iron site can activate the dioxygen. So, not only you have a vacant site over here, and this vacancy is taken up by the dioxygen molecule, and that dioxygen molecule, when it is attached, then only we get some internal electron transfer. So, and this sort of thing is very common, because in most of the cases what we have seen in case of cytochromes, that you can have something where the metal center is not getting oxidized, your ligand center is getting oxidized, and you get so many other products. That means you can generate a free radical system, and that free radical mediated reactions are very common with that of your iron site, which is a redox active metal center, which can settle between the +2 and +3 oxidation state. Similarly, some other type of system will find that copper can also play the same role to generate the radical site, and that radical site can attract the hydride, and the product is something different from there, and the reactivity pattern is little bit similar to this system. But in case of that intradiol cleaving agent, so we have the intradiol one, the simple difference is you have now the same type of peroxy intermediate, but in different mode of action. So, if you just write this hexa coordinated site like this, you have oxygen. So, you have a peroxy link, and that peroxy link is attached to our semiquinone unit with oxygen like this. So, in this particular case, you have the labeled oxygens which is coming out from outside. So, this is our peroxy intermediate, that peroxy intermediate, that means you whatever you have supplied to the system as dioxygen molecule, which is getting changed to a peroxide species, and that therefore giving some different product in this case, and where we get intermediate as a acid anhydride. these are the labeled one, and iron still changes either a hexacoordinated form, or sometime if this particular motif is not getting disturbed, the protein environment is such that these particular positions are not altered. So, you can sometime get a 5 coordinated intermediate on iron. So, this is OH or O minus. So, this particular part what is forming there is our anhydride intermediate. And the final step, which is very interesting, that now this in this particular case water is not attacking here, water will be here to break your anhydride link. So, this is our product, we all know now. This for intradiol clipped product is this one. And these are the two labeled oxygens. So, these two particular system, that means these two subfamilies. So, there is some difference between their environment, because we see that this can give you some different reactivity pattern. So, we have basically two subfamilies. So, one is responsible for extradiol cleavage and the other one is responsible for intradiol cleavage, but they are different. So, they are different in the sequence, the amino acid sequence. In amino acid sequence, their structural fold is also different. That means both tertiary and quaternary structures should be different and their corresponding pockets are also different around iron. You can have the nitrogen and oxygen bearing ligands, but the exact nature of these ligands are also varying, because with this generalized picture we try to focus our attention on 20 families. There are a large number of these groups depending upon the biological source what we have. So, one source to another, but broadly we can classify based on this. So, in one case to another you have a different ligand around the iron center, and lastly the orientation of the secondary sphere. That means, the hydrogen bonding interaction what the imidazole ring is showing through its hydrogen on the superoxide or peroxide oxygen, and the orientation of second sphere, which is important. Second sphere active site. So, we want to have the hydrogen bonding interactions, how we can stabilize this particular arrangement. Otherwise, this particular arrangement, that peroxide link is not so stable when it is bound to this iron. So, we need to stabilize with some hydrogen bonding donor atoms. So, with this information that in one particular system is cleaving catechol in 1,2 fashion and another case it is cleaving in the 3,4 fashion. So, people tried to have something, that means small molecule system, which can show its functional activity, that means the model system, how we can make in the laboratory, which can show this sort of reactivity, that means the catechol oxidase reactivity pattern it can show. So, one such example you can find where this particular case, that means you have the iron site and chelating ligand we always try to take, and what we are getting, that we are getting the reactivity pattern from these 3 positions or sometimes from the 2 positions. So, at least 2 positions should be vacant, which can be occupied by the molecule which will be cleaved, that means the catechol molecule. So, at least 2 of the positions must be vacant around iron, and you can have a particular ligand system which can bind all the 4 positions. So, one such example of this ligand is well known also, and it can be abbreviated as a Tpa ligand, which is tris 2-pyridyl methylamine, tris 2-pyridyl methyl amine. So, that means you have a tris 2-pyridyl methylamine. So, you have this nitrogen, that means the amine nitrogen in your hand, and you just functionalize it like pyridine, pyridine, pyridine. So, it is basically a tetrapodal ligand, we call it as a tetrapodal ligand. So, this can go, so this is the nitrogen, this is non-pyridine nitrogen. So, this nitrogen which can go for binding this way, and lastly the other one. So, you have a regular octahedral complex. So, mononuclear octahedral complex, and we just try to see whether this particular molecule is very much reactive or not. So, the compound what we can synthesize from here is Fe, Tpa, which is a tetradentate ligand, and your substrate is tertiary butyl. So, it is tertiary butyl, ditertiary butyl DBC, ditertiary butyl catechol. And since this catechol is there, and interestingly we will find that there is no phenol type of ligand. So, all of them are nitrogen which are neutral in nature. So, we basically try to go for where this particular ligand system can also stabilize iron in the low oxidation state, that means the +2 oxidation state. But here the synthesized complex is in the +3 oxidation state, and catechol is providing 2 negative charge. So, overall compound is cationic. So, when we allow this compound to react with the molecular oxygen, we get something, that means this is a typical model compound. If we find something, some product, that means once you generate the semiquinone form, and the radical form is sometime because of this corresponding electron donation from the tertiary butyl group compared to the normal catechol unit, the tertiary butyl substituted one will have higher stability in the semi-covalent form. The radical form what you generate over here, that will be stabilized. So, that is the reason why we are taking this substitutions, the tertiary butyl substitutions on the ring. So, you get basically two products from here. The anhydride one plus one cyclized product, because we are looking for something, but sometimes we get some other transform product as well, depending upon the stability of the intermediate what we can generate through this redox process based on the iron site. So, the iron is definitely a very good catalytic site in that way. This is the T-bube substitution over here, is a 5-membered ring cyclized product, and people have identified that, and at the end here you have the aldehyde function. So, this is one product. So, basically from this reaction, after characterization of all these, we get product 1 and product 2 on a model system. So, it is a non-biological system. You can make the compound in the laboratory and you say some thing where you can have the dioxygenase activity using model iron complex, which you can make in the laboratory. So, having known all these informations that this one particular iron compound can show something. So, what we can do next, that means the reactivity pattern for some other substrates or catechol type of other substrates or some other molecule, sometimes some amino acids having some aromatic ring or some part, that means we can go for the modification. If we try to see more, we just go for the ligand modification. You can change the ligand system, that means you can attach one such phenol unit to the substitute that pyridine unit or some other donor groups including sulfur or other 2 oxygen atoms. So, modification of this can lead to something where you can have some different type of reactivity pattern. So, this is all about your iron, that electron transfer proteins based on iron. Now, we will go for something where you have the copper proteins, and these copper proteins, they are of different types and they are present in biological system. So, they are type 1, type 2 and type 3, and we can analyze those sites. So, different copper redox proteins, because we are looking for some electron transfer behavior. So, these are electron transport protein again. So, the copper based redox proteins. So, like iron, so copper can also settle between the oxidation states like +1 and +2, the cuprous as well as the cupric. So, spectroscopic characterization, that which one is copper 1 side, which one is copper 2 side and which one is copper 3 side, that can be known if we study in detail the spectroscopic features. So, characterizations of these copper sites are of two types. One is spectroscopic characterization and another is structural one. So, spectroscopic characterization, when we talk about for these electron transfer proteins, because we are talking everything on electron transfer. Proteins. So, spectroscopic signature is one such that what gives you the color. What is the color of this system? So, they are all, most of the cases, blue in color. So, they are also known as blue copper proteins, and these blue copper proteins having a corresponding band at 620 nanometer. That is why it is colored blue, and its corresponding epsilon value is pretty high, and it can be categorized as a charge transfer transition, and therefore, it is a charge transfer band which is coming and is basically originating from cysteine sulfur to copper 2 plus ligand 2 metal charge transfer. So, it is LMCT type charge transfer. So, once we get that, this strong electrical absorption, so electronic absorption spectra immediately tells us that this particular type of species having a lambda max value of 620 nanometer and epsilon value ranging from 3000 to 6000. Mole inverse, centimeter inverse. So, immediately we will find that when we get this particular signature, we label it as a type 1 blue copper protein, or sometime we abbreviate as T1 Cu, and there are a large number of such molecules available. One is, they can be from plant origin, they can be from bacterial origin. One is our well known molecule plastocyanin. Then one is also well known is azurin. Then stellocyanin and amicyanin. So, in most of the cases you see the name is ending with cyan, which is a color. The level, the adjective for that is for the color, the cyan, blue color. So, this particular case, that means once you know the corresponding band position and its epsilon value, you can say that spectroscopically you can determine that the type 1 copper is there. And another spectroscopic signature is your EPR, because sometimes it is very difficult to identify the metal center first and what is the corresponding environment around the metal. So, EPR is the second information which can supplement your spectroscopic characterization for this one. So, these are basically a typical type of your EPR spectra for that, and sometimes we get the hyperfine one for the copper center. So, 3 line can be there. So, this can be splitted into 4 lines. And we label this is a very well known one picture for all the EPR spectra for copper. This is the G parallel region and this is the G perpendicular region, and for all these things we just report this value and this value with a reference of dppH. So, these values also tells us something, so long we are not knowing anything related to the structure of the molecule, what type of structure it has, that means this coordination number and the geometry. So, this particular one, if we know large number of synthetic molecules and we can correlate those with that of our synthetic molecules, that the EPR signature is this, and we have the corresponding splitting. So, small hyperfine splitting is there. So, this splitting basically, and this splitting are labeled as A parallel. So, we have small hyperfine splitting. In the parallel region, not in the perpendicular region. So, this color and the EPR characterization then gives us some information that since we are talking these all for electron transfer behavior, so they must have some typical E0 values. So, we go for another level of characterization of these molecules, we will measure the corresponding E0 value. So, in most of the cases E0 is measured at pH close to 7.0, and in case of azurine, say one such example, I will give that some list for that. So, in case of azurine it is 0.415 volt, and if we just compare this corresponding value with that of our aqueous copper to copper 1 potential. So, all we know is that what is our aqueous copper to copper 1 potential, which is copper 1 is plus 0.170 volt. So, you have a corresponding potential which is high compared to your copper center, which is aqueous copper in solution, that means any copper salt in solution. So, a relatively high reduction potential. So, it is relatively high reduction potential. So, this particular one, so that means you have something that where we need some extra potential value to reduce this copper 2 to copper 1 center, and that particular one when we give. So, stepwise if we find that, we initially find that you have this coordination. So, this particular information will give you that at least you have one cysteine sulfur attached to your copper site. Then you have this one is very close to tetrahedral or 5 coordinated geometry, and this particular geometry then, without knowing the exact structure of this particular site, gives us some idea that it has to be a copper center attached to one cysteine sulfur, and two of them are histidine nitrogen, and one is S-methyl, that means methionine, and a weak one oxygen from some carboxy end, say a glycine type. So, within this environment, what we get that this one, when this oxygen is not present, that means this particular coordination is pretty weak. We get a weak coordination from the oxygen, and overall you have a tetrahedral geometry. If we just consider that you have these 4 coordinations only, that means the copper in an environment of, that means simply we write sometime that the type 1 copper in blue copper proteins are 4 coordinated one, 2 of them are nitrogen and 2 of them are sulfur. So, it is a 4 coordinated system. So, this particular 4 coordinated system, when we go for corresponding determination of E0 values, that when this particular species go for the catalysis, that means the electron transfer catalysis. So, this is for type 1 basically, then there are some which are type 2 and some are type 3. So, in this particular case, when we have the type 1 case, so one such potential just now we have seen in case of azurine, which is a type 1 also and is close to 0.4 millivolt or 0.4 volt or 400 millivolt, but in other cases for system like Lec-11, one such example is Lec-11, their potential is pretty high. That gives us some idea that how far, like cytochromes, that potential can change. So, from 0.4 it can go up to 0.7, so which is 785 millivolt. And for the same Lec-11 is the multi-copper oxidase system. Lec-11 is nothing but there are so many centers, because in sometimes if the system is pretty complicated one, we get the type 1. So far we have seen only the type 1, which is the simplest one, and the type 1, the simplest possible one is your mononuclear one. Then you have type 2, where potential is around 500 millivolt, and type 3, which is close to azurine potential, which is around 400 millivolt. So, in all these cases like azurine, Lec-11 and all these, we see that there is a corresponding charge transfer transition, and the center, that means the copper center is there present, and this particular one, the copper one, is more strongly bound to these two groups, that means the histidine nitrogen from here and there. So, this trigonal plane, this is one trigonal plane. if we consider this weak bonding from oxygen, we can consider it as a trigonal bipyramid geometry. So, this is the trigonal plane. So, these bondings are pretty strong. So, that immediately tells us that because you have the cysteine sulfur and we have the charge transfer transition, and that immediate charge transfer transition in this plane, the trigonal plane, tells us that you have a very strong interaction between one of these sulfur, that means sulfur of cysteine, and that basically is responsible for your color, but your methionine sulfur is S-methyl sulfur and that interaction is not so strong. So, that is why one of the cysteine sulfur is taking this particular trigonal plane. So, the basal plane it is occupying, and this methionine sulfur is taking the apical site. And during this particular interaction, if we just find that you have this cysteine sulfur, so there is a typical corresponding covalency. The covalency is generated when we talk about that from the detailed calculations of all these molecules. So, if you have a typical orbital over cysteine sulfur and the corresponding one for copper, so you have a particular type of covalence is generated from that p pi of cysteine sulfur, which is hybridizing with your copper dx square minus y square. And from the calculations and all other spectroscopic parameter comparison, we will find that because these two are very much interrelated. So, about 38% of this bond, this orbital is participating with that of our cysteine sulfur, which is 42%. So, if we have some strong interaction for these two groups, so they are basically dominating your color and the reactivity pattern. So, when we go for this particular change, that means methionine sulfur is interacting with the copper center, and when we consider as a tetrahedral geometry, that means this geometry, if you just simply consider this, that you take out this oxygen and you remain with these two, but this particular geometry is not this geometry, is not a regular tetrahedral geometry. So, when this particular oxygen is removing, is a highly distorted geometry, and this particular in biology we will call in all these cases they are enstatic states. So, you generate the enstatic state by simply removing this weakly coordinating oxygen atom. So, if we just simply remove this weakly coordinating oxygen atom and this particular one, so your interaction might little bit increase from the out of plane methionine sulfur, and that out of methionine sulfur has some role to play, because this particular one is closer to the transition state. So, for electron transfer to take place, there is a corresponding transition state which is again highly distorted. So, for a typical or a rapid electron transfer, your coordination geometry or that particular molecular geometry is such that initially when we talk about this one, if you remove this oxygen, so after removing this we will end up with a geometry which is trigonal pyramidal. So, this trigonal pyramidal geometry will tell us that during the catalysis what is happening to the copper center, because why this copper is so unique and copper can function for a typical catalytic role. So, during catalysis two things are happening there, that means electron transfer is taking place, and all we know from Frank-Condon principle and all that your electron transfer is pretty fast, which is in the range of 10 to the power say 12 second inverse. So, during that transition all we know from typical coordination geometry knowledge that the copper 2 is all the time stabilized in one particular coordination geometry, either square planar or square pyramidal, but copper 1 will be stabilized in tetrahedral geometry. But if we want to have a very rapid electron transfer within this range, that means the electron transfer rate is very high, which is 10 to the power 12 second inverse, we are tagging something, that means we are looking for that electron transfer for a corresponding conformational change around copper. So, you will have a conformational change and which is also taking place, but it is little bit slower compared to your electron transfer rate, which is 10 to the power 8 second inverse. So, what is happening that during the electron transfer, so one particular conformation is getting changed to other conformation. So, for typical or standard coordination compound, what we know, for a typical coordination compound, what we know that if you have a corresponding copper 2 compound or a copper 1 compound, what we find that copper 1 will be stabilized by low coordination number and sulfur type of donor groups can stabilize the copper 1, but the copper 2 is stabilized by some other different compound. So, if we see that for a typical coordination compound synthetically prepared in the laboratory and we look for some electron transfer, and if we try to isolate both the compounds, it will be very difficult for the same ligand environment to isolate the copper 1 compound as well as the copper 2 compound, because in case of copper you will be losing some donor atoms, you will be losing some ligand groups, and in copper 2 it will attract more ligand groups. Suppose copper can be stabilized in a coordination number of 2 or 3 only. So, when you oxidize a copper 1 compound to a copper 2 compound, what will happen? It will try to attract, because you are just oxidizing the center, it will be electron greedy and it will attract more ligand centers. And if those ligands are not available, like your biological system, the biological donor atoms are not available, in synthetic molecules it is not available, the suitable ligands are not available, what it will do? It will simply attract the water molecules or hydroxide groups. So, it will stabilize the thing in higher coordination number, but that is not happening in all these biological system. So, you center should be protected such that you will not have some vacancy, that when you move from copper 1 to copper 2 compound, no water molecule should enter to this particular site. So, no water, no hydroxide group can enter to this particular site. So, this copper site is highly protected. So, we should have some intermediate coordination geometry, which is your transition state. So, in the transition state we will have a typical coordination geometry, which is neither a tetrahedral geometry nor a square planar or a square pyramidal geometry. So, that is why all these copper 1 type of molecules we have a highly distorted system, and that distorted system, one such example for that is your trigonal pyramidal coordination. If you just simply go for this trigonal pyramidal geometry, which will be favorable for both copper 1 and copper 2. So, this is the basic idea behind doing all these studies that it is favorable for both copper 2 and copper 1. So, if we get some intermediate geometry, then from this trigonal geometry, if you go for copper 2, then it simply goes like that. It will attract some weak interaction from this oxygen like water. This glycine interaction, why it is coming? Because it is in the +2 oxidation state. So, when it is in the +2 oxidation state, it will attract some water molecule or hydroxide such that it can move from a 4 coordination site to a 5 coordination site. But when it is not there, when we want to stabilize it, so it is in the trigonal pyramidal one. So, after that electron transfer, so when you have the electron transfer, because sometimes electron transfer is taking place after your conformational change, or sometimes after electron transfer you have the conformational change. So, when you have a intermediate arrangement, that means it is very easy to go from this intermediate arrangement to a tetrahedral arrangement with a minute adjustment, with a very small adjustment from a trigonal pyramidal arrangement, you go for a tetrahedral arrangement, or in other case a square planar or square pyramidal arrangement. So, it will attract more molecules or more groups, and the simplest possible one is your water molecule. So, there are plenty of water molecules available in the system, and that basically tune this particular coordination geometry, and when we directly measure it, in our next class we will see that when we just simply measure it for its different E0 values by looking at their corresponding cyclic voltammogram. So, for this cyclic voltammogram, the pattern of those cyclic voltammogram will tell you that whether there is a typical conformational change or not during this electron transfer. Suppose we are going for a copper 2 plus to a copper 1 plus reduction. So, this basically a cyclic one, and when both the geometries are interconvertible, then only you get a corresponding very symmetric cyclic voltammetric response. Otherwise, your response will not be symmetric in nature. So, if your measurement will tell you that some asymmetry has been generated for this cyclic voltammetric pattern, you can say that during that electron transfer your conformational change is taking place. So, one particular geometry is changing to the other geometry. So, that we will see in the next class. Okay. Thank you.